Hi everyone, first I want to thank the organizers for the invitation. It is a great pleasure to share my work with you today. My name is Luca Pinello and I am an assistant professor at MGH and Harvard Medical School. And today I want to talk about quantification of editing events at off-target sites with CRISPResso2. Okay, let's start. But before, a quick disclosure. I am a co-founder and shareholder of Edilytics. Okay, this is the outline of my presentation. I will first present strategy to select regions to investigate off-target activity. Then I will introduce CRISPResso2, a tool for the quantification of editing events from next-generation sequencing data. And then we will explore two strategies to validate off-targets, targeted sequencing or whole genome sequencing data. Okay, why do we care about off-targets? Well, when we perform genome editing, we want to design and use guides that are efficient at the intended on-target site, but we don't want to have modification in other parts of the genome. This is really important point for safety of genome editing intervention, and several strategies have been devised to predict or detect off-targets event, as you can see from this picture. So now, regardless of the strategy we end up using, usually there are two ways to validate off-target sites. One is to perform targeted deep sequencing of sites obtained by in silico prediction or experimental assays. The other is to profile the entire genome in an unbiased way by whole genome sequencing. In theory, this strategy is unbiased, however, often we don't have the required coverage to detect rare events. Also, this strategy is not cost-effective. Okay, now with this in mind, let's briefly introduce the techniques to discover off-targets. First, we can use in silico approaches. The basic idea is to enumerate all the sites in the genome that have a similar sequence of the selected target. Some models improve on this simple enumeration using additional information, like enzymatic modeling or more powerful machine learning models, like random forest or deep learning. In general, for this model, it is important to consider, first, the number of mismatches that we want to tolerate, usually up to 4 mismatches, given that there has been shown that off-targets with more mismatches are rare and with lower modification frequencies. However, they can still happen, so we cannot ignore them. Second, we need to consider sites corresponding to DNA or RNA bulges, even though these events are rare. Finally, for a comprehensive search, it is paramount to include personal genetic variants, since this can dramatically change the on and off-target landscape. We discussed this in detail in a paper that we published in 2018 in the CRISPR journal. And you can read this paper if you want to learn more. So now, despite there are several tools to enumerate off-target in silico, they don't offer an enumeration based on all the 3 points I just described. To fill this gap, we developed and recently released a tool called CRISPResso2 in collaboration with the University of Verona. This tool efficiently enumerates target and potential off-target sites and importantly support searches with mismatches, DNA or RNA bulges, and also genetic variants. In addition, we also provide information on the enrichment of off-targets in different functional annotations, like coding regions, and non-coding functional annotation like enhancers, promoter, and insulators. As you can see from this plot on the left, the relative increase of off-targets when accounting for genetic variants is dramatic, and also different for different annotations. So we really need to account for genetic variants to have a comprehensive enumeration. If you want to use CRISPResso2, the software is already available, open source and free to use. And if you want to learn more, you can read our manuscript that was published last month in Bioinformatics. In addition to in silico methods, we can also discover off-targets using in vitro approaches. In these methods, we first extract the cellular DNA and then after exposure to the nucleus and the guide, different strategies are used to enrich and sequence only the cleave fragments, like in circle-seq or size-seq. Alternatively, we can sequence the entire genome and look for specific patterns in the sequencing data. So these methods are in general highly sensitive, however, they cannot take into account endogenous DNA repair mechanism nor cell type specific chromatin structure. Also, they don't recapitulate in vivo concentration of nuclease. Finally, there are also methods to detect off-target events in cellula that account for cell type specific repair mechanism and chromatin architectures. These methods are based on different ideas. After transfection of the nuclease and the guide, we can detect the editing based on different strategies. For example, in guide-seq, cells are flooded with short double-stranded oligodeoxynucleotides that get integrated at double-strand break sites and that are used for targeted amplification. Bless and Bliss instead exploit biochemical tagging, and Discover-seq instead uses immunoprecipitation of a protein that binds double-strand break sites in cell. However, these methods have also some drawbacks. For example, toxicity in some cell types for guide-seq and limited sensitivity for Discover-seq. Importantly, the number of sites recovered with these different approaches is dramatically different, and in silico methods report too many sites, many of which are false positive. So a common strategy is therefore to combine the strengths of each strategy to create a list of sites to validate. For example, in this recent paper from the Young Lab, they use up to 3 mismatches in silico and their sensitive circle-seq assay to create a list of sites to then validate in vivo. So now for the validation there are several strategies like T7 Endo or Surveyor that are gel-based, or analysis of traces from Sanger sequencing, or direct detection of mutations using next-generation sequencing. And we will focus on this strategy. Importantly, next-generation sequencing is currently the gold standard for confirming and validating off-target events. Okay, so assuming that you have selected and sequenced your sites by next-generation sequencing, how do you analyze the data? Well, for this you can use CRISPResso2, a tool that is specifically designed to detect genome editing events. This tool was developed by Kendall Clement, a very talented postdoc shared between my lab and the Young Lab. Briefly, starting from sequencing reads, a guide RNA and a reference sequence, reads get aligned and classified in modified or unmodified. Finally, a summary is created with several informative plots, so you can assess editing rates and outcomes. And this tool, was published last year in Nature Biotech if you want to learn more. Now, when we sequence our regions of interest, there are like two common strategies. The first one is based on amplicon sequencing, where specific primers are designed and used to amplify the region of interest. The second one is based on sequencing the entire genome. CRISPResso2 supports both scenarios. So how do you use, CRISPResso? Well, you can use CRISPResso online through our website, available at CRISPResso2.pinellolab.org, or using the offline version with more options, available in Bioconda or as a Docker image. Importantly, CRISPResso can run in any modern operating system, so you have plenty of options. Now, depending on your sequencing strategy, we have specialized tools. However, all of them share some important steps. The first one is quality filtering to remove low quality reads. So the second one is adapter trimming. Then we have read merging if used parent sequencing. And then we have an alignment procedure optimized for genome editing outcomes. And finally, the visualization. So for targeted sequencing of a single amplicon, you can actually use the website. Okay, let's take a look. So this is the website interface. It's quite intuitive and simple. You just need to select the editors you are using, provide the guide that was used in the experiment, upload your sequencing data file, and finally click submit. After you click, a nice report will be generated. Also, on the left of the page, there are several examples for different applications, including base editors, HDR, or at least specific quantification. So you can quickly check what is the required format and what is the expected output. So now let's take a look at the report. First, you can get information on QC and the alignment statistics. Second, you get a quick summary of editing frequency at the site analyzed. If you don't have off targets, here you should see a number close to 100% for the unmodified category. Next, we show a per nucleotide summary for the editing events, so you have an overview of what happened to your site. And here instead we show a positional profile for insertion, deletion, substitution separately and across the entire amplicon. The expectation here is to find events close to the expected cleavage position that is highlighted here with a vertical dotted line. Finally, we can go even deeper and decompose these profiles based on the alleles recovered in our experiments and report their frequencies. This is very helpful to spot potential artifacts or uncover genetic variants that were not present in our provided reference sequence. Importantly, in CRISPResso2 we have several tools to compare treated samples with control samples. Here you see on the left two samples overlaid in the same plot, one treated and one control, while on the right we consider their difference. It is clear from these plots that the experiment worked well, however you can immediately notice some spurious signals related to sequencing errors. So, bottom line is like always perform a control experiment to have confidence that what you observe is real. Going back to the tools available in CRISPResso2, I want now to talk about CRISPResso WGS. This tool can be used to analyze WGS data as the name suggests. In CRISPResso WGS, given a BAM file and a set of regions of interest, for each region we first extract the reads that fully cover it and trim them. Then based on the obtained reads, we run the same CRISPResso analysis. Here we do this to make sure to have the same amount of information for each nucleotide and to provide an accurate estimation of editing events. However, there are several challenges when using WGS data. First, off-target events may be rare, and given that WGS data have usually lower coverage compared to amplicon sequencing, you may not detect them at all. Another point to keep in mind is that when the events you want to detect may be recovered by only 1 or 2 reads, a sequencing error of 0.1% can create a lot of false positives. So to improve the quantification, we are currently exploring the usage of predictive models that have been recently proposed to increase our sensitivity and reduce false positive. For this task we're using in Delphi a predictive model that, based just on the sequence of a guide and of its target, can predict the expected repair outcomes and their frequencies with high accuracy. So the idea here is quite simple. Given a guide, a genomic location, WGS data and a reference genome, we first run CRISPResso WGS to obtain a table with all the alleles and their frequencies. In parallel, using the sequence of the guide, we can predict which alleles we should expect to see in our experiment, based on the in Delphi. Based on these two tables and correcting for the used WGS coverage, we can then define a concordance score. This is important since, as you can see in this example, some indels are supported only by 2 reads. So it is easy to get, false positives. Okay, is this working? Yes, it does. So based on this strategy, we try to rank all the sites with up to 2 mismatches based on the concordance score just defined and the editing frequencies for a given guide targeted VGFA and for which we had guide-seq data. As you can see in this plot, the top site is the target site, not surprisingly, but this is followed by sites that have been validated by guide-seq. In addition, for each site, we can also inspect what are the alleles that are driving our concordance score. So as you can see here, you have like, the tables for this particular site, and also below you can see all the alleles that, we recovered and match our prediction. So now we have a nice strategy to unbiasedly look at the entire genome if you want. So this feature is still not available in CRISPResso2, but we hope to release a software soon. So if you're interested, please contact me offline, and we're happy to share this with you. So in summary, it is important to select off-target sites for validation based on in silico and experimental data. Second, CRISPResso2 can be used to efficiently quantify editing event at on and off-target sites, and also targeted versus genome-wide characterization can be used with it. However, as I show you, targeted sequencing is more cost-effective and more sensitive, and also WGS data is not truly unbiased, given that rare off-targets would be missed based on the typical coverage that we can currently use. And finally, always perform control experiments, because there are a lot of artifacts that, depends on the technology that you use, also on the site in the genome. So always perform control experiments. Okay, finally, I want to acknowledge my lab, in particular Kender Clement and Jikong Lin for the development of CRISPResso and for the WGS analysis. Also, I want to thank Kit Zhang, David Liu, Ollie Rees, and Daniel Bauer for helping us to create CRISPResso, and also the University of Verona for helping us to create CRISPRids, a fantastic tool to in silico enumerate potential off-target sites. Also, I want to acknowledge my funding sources, DARPA, CZI, and NIH for supporting my lab. And with this, I will stop here and I will take questions. Thank you.